Okay, let's make a start. Hello, everyone. It's Kelly from the Doctoral College. I'm the Events and Communications Officer. And welcome to the Doctoral Inaugural Lectures, whether you're watching live or on the recording. Thanks for taking the time out to spend it with us. And I hope you're enjoying the sunny weather if you're in the UK. Welcome to the Doctoral Inaugural Lectures. I hope you can all see the screen. Yeah, that looks pretty good. I'm just going to say a few things before we pass over to the speakers. The session will be recorded. You can follow up again if you'd like to. Your mic and your video, if you're an attendee, should be turned off as you arrive. If you do have an issue during the session, please do use the chat function not to interrupt the speaker. I'll be on the chat function, putting some little bits of interest comments on there as well. But if you have any problems or anything, then just add it on there and I'll pick it up. Feel free to ask questions in the chat function as well. If you would like to ask questions during the Q&A personally, then I can unmute your mic. Just let us know. There's a couple of things with Teams. You are able to switch between a presenter and a presentation once they share their PowerPoints. And you can do that by clicking on the box which you are most interested in once we're going through the event. I'll put all this information in the chat as well so we can refer to it. If you want to pin a video to focus on a particular video, you just right click it and click pin and it will stay on your screen. And you can pin as many videos as you want to fit on your screen. And then if you change your mind for anyone, then you can just click unpin. And also, if you want to reframe a video, Teams can crop videos a little bit sometimes, which can make it look a bit strange. If you want them to fit in your frame better, then you can select fit to frame and see the entire video. So that was just a few bits of logistics before we get started. I'm now going to pass over to Professor Dave Lambert, who is Dean of the Doctoral College. He's going to introduce our first speaker.
Thanks, Kelly, and welcome everybody to the Doctoral Inaugural Lectures, which is part of our research showcase that we've been running today. These lectures really are, for me, one of the highlights of the academic year, that it gives me an opportunity to hear the real best of the best. I get to do all the nominations of examiners and read all kinds of reports and this, that and the other. And this really puts a real face on excellence. And I think excellence is a word that I really want to emphasize. It's incredibly prestigious to be selected to give these doctoral inaugural lectures. So, I can speak specifically for College of Life Sciences, which is my own college, is that there is an intense competition to get down to the final 2 or 3 who will give presentations. So this is a very prestigious event, and the speakers are to be commended on the quality of their work to get them to this point. So we're really looking forward to hearing what you have to say. So the first speaker today will be Dr. Rob Wilks. So Rob obtained his PhD in 2020, I think it was. And he's currently Senior Lecturer in Legal Practice in the University of South Wales. We have also on the call, and you should be able to see in the bottom pin of your screen, we have 2 BSL interpreters. So a very warm welcome to Julie Doyle and Stephen Bratton-Wilson. So you don't want to hear me prattle on. I think I'm going to hand over to Rob. So Rob, please give your doctoral inaugural lecture, Making Equality Law Work for Deaf People. Over to you.
Thank you very much, David. I'm just going to share my slides, so please just give me a moment and I'll get myself organised. It's uploading now, so hopefully any minute you should see it. And whilst it's uploading, I thought I'll take the time to introduce myself. Yep, I'm indeed Rob Wilks, as already introduced. Fortunately for you, I've brought along 2 BSL interpreters for you today, so you can understand my sign language. I'm obviously a deaf person, which is why I'm using British Sign Language today. I was born deaf, and so have been all my life. And I very much appreciate being invited here today to deliver to you the conclusions of my PhD thesis. Now, to do that in 20 minutes is very difficult. Just talking about my life and my experiences is a challenge in itself, and fitting that in. So hopefully this will work. I'm sure somebody will tell me off should I start to get too close to the time. Hopefully you can all see my slides now. I'll make a start. So the title of my thesis is Making Equality Law Work for Deaf People. And as David's already said, I did finish it in January 2020. And I'm unfortunately still waiting for the opportunity for my new style, doffing the cap and gown, to be honest. I'm very keen to wear it. So I'm very excited about the day comes when things are back to some kind of normal where I can graduate. So on to my presentation. The first thing I would like to talk about is that I'm aware that most people who have heard my thesis for the first time have probably never come across anything to do with deaf issues before. So I started the PhD with that kind of framework. So I did go into explaining a lot of concepts at the beginning, really, to talk about deaf people, and then moved on to equality and then the law itself. So the first thing I did was obviously take a look at what was out there already, and dealt really with three categories: the hearing world, the disabled world, and the deaf world. Now, I've split them into society into three categories because I was asking a really big and important question, and that was, where do deaf people fit in? In the hearing world, I won't talk about that too much because you all live in that world. You are people who hear. You know what that world is like. That is your normal. Society is very hearing based. It's audio based. You communicate with each other orally. You listen to radio, you listen to each other, you listen to the television, you imbibe sound, you imbibe information through sound, basically. So I don't need to go into too much detail, that's your lives. So then we'll move on to the disabled world. Now, there are a group of people in the world who classify themselves as disabled. Now, what that means, there are various models that explain what that might mean. So first of all, the medical model, most legislation and most of society focus on this model, the medical model of disability. They see disabled people as having something wrong with their bodies. For me, people would say my ears don't work, basically. Wheelchair users, people would consider their legs not to work. They have mobility issues. A medical model sees that through that lens, and legislation would also do that. There are other models which are now more predominantly popular in the world at the moment, the social model of disability. So that is opposite to that, and that model basically would say that actually, rather than the physical issue being the problem, it's actually societal's problem. So it's society that has the problem. Society has created barriers for disabled people. So the onus is on society. Society disables people. And then there is a minority group model. And here it's disabled people who would have a shared experience of oppression, for example, would want to be seen through this lens. A minority group comparing themselves to other ethnic groups, for example, or other language groups. And there is also a cultural model, and some academics argue that disabled people are actually a cultural group of people that have a shared cultural value and beliefs amongst them. So the problem with that model, however, would be, well, as deaf people, we might be seen as part of the disabled community, but other members of the disability community are hearing and don't sign. So there is still a barrier in place. And we have a similar issue communicating with disabled people as we do as hearing people have difficulties with us. So there's a communication barrier there. So that's just a flavour really for you. And the deaf world's quite interesting because I presume some of you, or most of you, might never have considered this before. Deaf studies have undertaken for decades now an argument that deaf people are a cultural linguistic group. What defines us is not our difficulty in hearing or anything, but what actually defines us is our shared language. the interesting thing is globally, there are various sign languages all around the world. It is not universal. It's not homogenous, for example. It's not a shared language. Each country would have its own signed language, indigenous to their own country. Within those countries, there are dialects and accents. So the way I'm signing, I actually have a Welsh accent of British Sign Language, and I would sign slightly differently to Scottish deaf people and deaf people from London. So we are a cultural linguistic group, and that's what defines us, is our language. And also, if we look as well at sign language people, Paddy Ladd, Define, Gulliver and Bansbury all say and have argued that actually deaf people are part of signing people. We are including people who use hearing people who sign too. So that could be interpreters or coders, which are children of deaf adults who can hear. There are those people who have learned to sign later in life. So sign language people are people who have learned sign language, whether they're deaf or not. And I would argue that deaf people can be classified as an ethnic group. And there is case law under the Equality Act around that. I feel that most of the criteria we would meet. So I started my thesis with an explanation of these various groups of people in society, the differences between hearing people, disabled people and the deaf world. Now, just trying to make sense, I suppose, of all this information here now, we need to look at identity in order to do that. So in order to define who we're talking about, and it's important that we see the application of identity to the law. So I've used me as an example. So let's have a look then at different parts of my identity and what's protected by the law and what isn't. And it's quite clear from this slide here that, oh, by the way, if you see deaf with a small lowercase d, that is a medical definition of being deaf. And that's protected under equality legislation, under disability, disabled, I'm white, British, Welsh, I'm a husband, we've got that covered under race. I'm a husband, which means that I have protected characteristics around my marital status. As a father, I have employment rights, so that's social welfare, paternity. And as a lawyer and as an academic, employment law. I would say that the most important part of my identity is being culturally and linguistically deaf, hence it having a capital D here, but I'm not protected. That part of my identity, the largest part of my identity, is not protected, and that's a difficulty for deaf people. So just to sum up this point really, what's happening here I've defined as the deaf legal dilemma. As you can see from the other list of identities, those intersectionalities of myself here, that most of my identities are actually protected except for my culturally, linguistically deaf side. So I have to, in order to access my rights, I have to label myself as disabled differently so I can gain some reasonable adjustments from my employer, for example. I have to then identify myself as disabled, even though I might not do that. I personally don't have a huge problem with that because it is possible to have multiple identities. That's fine. The issue we find is my cultural and linguistic identity as a deaf person is not protected. So therefore I don't have any rights because of that. And really the disabled label doesn't recognise my deaf heritage, my culture, my language. And surprise, surprise, a lot of deaf people don't want to be cured, actually. You might find that surprising. If a lot of people would say if there was a cure to be able to hear tomorrow, they wouldn't want to because it's very much part of their identity and it's who they are. My thesis talks about how deaf people have experienced barriers and inequality because as a deaf person, as a cultural and linguistic deaf person, we are not covered by legislation and the law. And so we have to use other identities to access our rights. So my PhD is obviously law-based, and therefore I had to study the law and had various options that I could consider. And you've probably worked out already that it makes sense to choose equality law to look at. Yeah, it makes sense. We're talking about inequality, so we'll use inequality law to look at why things don't seem to be working for deaf people, why the law doesn't work for deaf people. Equality law has a lot of crossovers with human rights law as well, as we know, so I picked both of those to focus on, and hence my PhD being based here. So the first step for me was thinking, okay, as academics, you know, we really love to categorise everything as much as possible to make sense of everything. So I needed to make sense of what I was researching. So I needed to do that before I could even begin exploring the law itself. So the big question here was, what is equality? And when I delved into that, I remember thinking at the beginning that it was an absolute minefield. I cannot believe how many definitions of equality there are and how many concepts there are, how many different ways of viewing equality and various approaches. It actually gave me a headache for 2 years, I have to say. It was a minefield. So I then started to try to get to grips with this and make sense of it. And Vickers has said that there are different analyses of equality that may be suited to different protected grounds. And here, that made sense to me, that resonated with me, and it made a lot of sense. Fraser also gives some examples. Fraser felt that class inequality was best understood in terms of redistribution. And so sexual orientation inequality was best understood in terms of recognition. And so that made some sense. And then I thought, OK, so which inequality concept would suit the deaf world? And that was my starting point. So along that particular journey, I came up with 3 categories. I say I developed them. I'm lying here, really. I didn't develop them. I actually, should we say, borrowed them. And so the 3 are formal, substantive and transformative. Now, I'll just go through very quickly what it means, and you can probably see here by the images that I've demonstrated for you. You can see the differences. So we have formal equality, which is all about treating everyone the same. Now, as you can see in this particular situation, there is a beautiful view beyond that fence. Now, there are 3 people of varying heights. And the fence is the barrier. They all want to see that beautiful view. Now, the shortest person is unable to see because the fence is in the way. But formal equality would say, well, we're treating you all the same. So you can hence see the results and the issue here. That person still cannot see. There is no disadvantage that has been removed, unfortunately. But we're treating everyone the same. Now, when we move on to substantive equality, we're talking about removing the disadvantage. So we've given them blocks to stand on. So there you go. You can all now see the view. Now, when they go home, those blocks will probably be taken away and put in the shed, never to be seen again, gathering dust, until the next person who needs it. And then they might be brought out if you're lucky. So that's a temporary fix. Yeah. So then we move on to our third scenario, transformative equality. Do you know what? The fence is the problem. Let's just get rid of the fence. Why is that so difficult? And I would say a lot of public bodies, legislation that create equality law don't think about transformative equality enough. This could solve a lot of problems for minority groups, especially deaf people in the context I'm giving you today. So I have 3 categories. And so what I've started to do is to think about applying concepts to these. So first of all, I needed to identify which concepts are most suitable for using with deaf people. And Vickers, as I mentioned earlier, had talked about various concepts being more suitable, various lenses being better to look at deaf people through. So I looked at literature and looked at the concepts of equality, then which ones were talked about the most. And there were three. So there was dignity, social inclusion, and challenge to oppression. Challenge to oppression in the deaf world is called audism. So these are our concepts of equality. Fantastic. And also, equality of opportunity is also popular amongst academics and would apply to most groups. So I also used this. So 4 in total. So where do those concepts fit in to formal, substantive or transformative equality? As you can see, the results here quite clearly. And I thought of these as deaf equality concepts. And I've done what Vickers has suggested, thought about which concepts apply to particular groups of people. So it's certainly worth thinking about the concepts of equality in bearing that in mind as we go through. So my PhD obviously is in law, so I had to look at the law and was finally able to do so at this stage. Actually having a look at the law. I'm a qualified solicitor as well, so this is great. I grabbed the opportunity to move away from philosophy and now look at more of the nitty-gritty of the law, which is where my comfort zone is. So a few things to talk about here. It's how equality works for deaf people. How does equality law work for deaf people? So having a look at 3 things. So I looked at European Convention on Human Rights, the Equality Act 2010 and the Convention on the Rights of Persons with Disabilities. So I'm aiming at this point now to identify which concepts that we've already talked about fit in with each of these, and also to decide whether they're formal, substantive or transformative. The European Convention of Human Rights, I would argue, is linked to the concept of dignity. It's about preserving human dignity, protecting our rights in this manner. So the only issue I had was the way that it works is very formal. So there is no direct system of enforcement, which I will go into a little bit later. Now, obviously, the convention is relevant to deaf people because it gives us the right to a fair trial, freedom of expression, and so on, as it does with everybody else. Now, the most relevant article is Article 14, which talks about protection from discrimination. Now, most of you as lawyers would know that Article 14 has limited rights. It only comes into force if one of the other absolute rights are also used. Okay. Hence, we have an issue. So we have, in order to claim against discrimination under this convention, we have to do that if your right to life, for example, has been infringed, or if my rights to expression have been compromised. So enforcement is an issue because it's under the individual enforcement model. And this means we need to rely on individuals to bring the claim forward. And as you know, in the UK, we have to go through our domestic hierarchical legal structures first before we can take this on to the European Convention. That takes a lot of energy, a lot of effort, a lot of money and time. And because of that, it seems like there have only been 4 cases relating to deaf people that have come forward. So let's move on then to have a look at the Equality Act 2010. Now, I was quite shocked with this because I actually saw there was a mixture of formal, substantive and transformative equality here. The formal aspects are more things like this direct discrimination. So indirect discrimination, the usual discrimination provisions. But I feel reasonable adjustments are more relevant to deaf people here. And I would say that is substantive because there's an aim to remove disadvantage here. Now, there isn't much case law in relation to deaf people here, so I had to look wider to disabled people who are making claims for reasonable adjustments. And it seems like, again, it's a single one-off adjustment, then you're more likely to be successful as opposed to a recurring reasonable adjustment. And guess what deaf people need? Recurring adjustments. Every time we come across people who can't sign, we need interpreters. So that is a recurring cost. And that could be why deaf people are still facing inequality and barriers, because of the recurring need for a reasonable adjustment. Wheelchair users would need a ramp or would need access into a building. That's more likely to be seen as reasonable because it's a one-off cost. And so there's quite interesting examples around case law with that. Public sector equality duty. So that's another aspect of the Equality Act. It's actually quite transformative. I would say this removes barriers. It really does try to change society's attitudes towards disabled people, deaf people in sign language, so potentially quite transformative. So moving on to convention here. This is potentially transformative as well. I've actually talked about, it actually talks about national sign languages in the convention, which is fantastic, and that has huge implications for the deaf communities. Again, the convention relies on the individual enforcement model, so you have to go through your domestic court hierarchy as well, as I mentioned with the convention earlier. And so that means the same issues apply: time, money, effort, all of that. It's interesting, however, because the system here is complaint-based. So individuals, when they've finished going through the domestic systems, they can then take that to the Committee for Human Rights at the UN, and they will then look into that. There have been 19 complaints at the time of my PhD, and there were 19 complaints to date, and 3 were in relation to deaf people. And quite interestingly, one was an Australian deaf person, and they had really much stronger advocacy networks there. But this is an example of transformative equality. So we can see various issues that we might explain as to why deaf people are still facing barriers and inequality today. So I'm a lawyer, right? I like solutions. I want to look at how we can get past this. So the deaf community have been campaigning now for many years for a British Sign Language Act to protect our language. And that is a solution, and that would deal with the cultural, linguistic, deaf community being protected. It would consider sign language, which is a huge part of our cultural identity as a deaf community, and that would help solve that problem. Of course, we don't have a BSL Act in the UK at the moment, so we have to look elsewhere. I did look to Scotland. Now, Scotland, fantastic, have actually appeared here: New Zealand, Finland and Ireland. So I've looked elsewhere. I won't go into too much detail on each country. I don't have time, unfortunately, to do that. I'm sure you can take a look yourselves. You're more than welcome. But basically, it seems like this is actually quite transformative. The problem we have is there's not enough empirical research to date about the impact of those signed languages being recognised formally. And in New Zealand have undertaken a review. However, it only scratches the surface. It doesn't seem, it's more of a focus on service provision. So it's about looking to see whether they're complying with the Act in terms of their services, rather than about how the impact on deaf individuals and the deaf community. The deaf community, they did say in New Zealand, do find they have more pride in their language and culture, but that wasn't defined as to how that manifested, how that looked. Is there a change in their everyday life? And so there is huge gaps in the research, and that's where it came to an end, really, and that's where my research had to stop there. So the aim, I would say, to continue is to look at further impacts of the recognition of signed languages. So when it comes to the UK, just to finish off, the UK government, Conservative government, have made it very clear that they're not interested in recognising British Sign Language and having a BSL Act. And you might have heard about a campaign on social media called Where is the Interpreter? You know, we have the UK briefings, coronavirus briefings, with Boris Johnson. You never see a BSL interpreter next to 2 meters apart from the Prime Minister. Wales, Scotland, Northern Ireland have sign language interpreters in the room. Now, the UK government will not do that. They use the excuse that the room isn't big enough. Well, they have been using that excuse. However, as you know, this week they've moved to a delightfully new, very posh 2.6 million, wherever it was, pound refurb. A bigger room, and still no BSL interpreter in the room. So the Conservative government, less said about that issue, I think the better. But in Wales, there has been some discussion around creating a BSL Act in Wales. Ireland are moving ahead with theirs. Obviously, the devolved nations are moving ahead with this, so why can't the UK government? Our devolved countries are leading examples at the model, are role models for the way forward. So to conclude, I have explained to you why deaf people have to identify as disabled in order to access their rights. And this is our deaf legal dilemma. I've talked about the relevant equality analysis that I've given to the deaf world and all the various concepts and relating that to our deaf world. And I've developed 3 categories. I call them precepts to help us make sense of those concepts and how they work in practice. And it's quite clear that formal substantive equality isn't quite doing it. It's still creating barriers for us. It's not solving our problem. Transformative equality, on the other hand, could be our solution. And we need to see more of this. But there isn't enough transformative equality in the law to make it transformative. So we've applied these precepts and concepts to the law and to deaf people, and we've explored the idea that what could be very transformative would be the recognition of British Sign Language and creating an act. I started, I'm obviously a lecturer, and my main function is obviously to teach, and this year has been pretty overwhelming. You know what it's like, moving everything online, as well as the usual stresses of teaching. But I'm making up for it now in the summer term as things ease off a little bit. And I'm going to be doing some research on the impact of education of the BSL Scotland Act with Rachel O'Neill at Edinburgh. And so I'm going to keep an eye on Ireland. They're moving ahead. They're having a review soon, and Scotland will be reviewing their sign language acts because they were recognised in 2015, so they're due their review. And Brazil's very exciting too. And I say that because they have recognised their national sign language. And I think that's in 2003, 2005. And the number of deaf people with degrees and master's degrees and PhDs have escalated as a result of that, which I find quite fascinating. So I'm planning at some point when I get some future funding to do some further studies into those other countries I've mentioned and looking into deaf communities' views and perspectives on the recognition of their sign languages and to see whether it's made a difference in their lives or not. And I think it's time for me to now stop and to say thank you.
Thanks very much to you, Rob. I'm sure you'll all agree that was a very interesting lecture. And I'd also like to thank our interpreters too. I had no trouble understanding the whole thing. Absolutely outstanding. We've got time for a couple of minutes of questions if anybody wants to ask anything. If not, I'll just plough ahead because that's what I do. Does anybody have any questions that they'd like to ask? It might be difficult for me to identify you, but any questions from anybody? Okay, I'll ask a question then. I was particularly interested, Rob, in the let me just say, you made a comment that deaf people should be considered an ethnic group. I wonder if you'd like to expand on that a little bit. I find that quite fascinating as a concept.
Yes, I can do that. Put me on the spot now and try and remember the arguments for deaf people being an ethnic group. There have been cases that have looked at certain kind of the criteria for identifying an ethnic group. So there's things like shared history, there is shared language, shared religion, for example. There's a whole number of criteria. I think you could argue that deaf people as a community group might share most, and even religion, because there is a sense of a shared religion within the deaf community. And we can argue about what religion means, but it's more about the way that deaf people would practice religion as part of their deaf culture and their deaf identity. So it's not necessarily important which religion we discuss, it's more about how they practice their religion as part of that wider group. Because there is a deaf church, for example, there are lots of churches that might have communication support workers who would come in and interpret church sermons, for example. So there is communication support services provided in church. So we could argue that even religion within that wider group could help to class deaf people as an ethnic group. So yeah, just off the top of my head, without my thesis being available at hand, hopefully that's answered your question.
Yeah, absolutely. That's really, as a concept, I think that's really interesting, and it certainly made me think. So does anybody have any other questions? Any other questions from the floor? So in the absence of that, it just remains me to thank you again, Rob, and to commend to you our Doctoral Inaugural Lecturer, Dr. Rob Wilks. Thanks, Rob. What would happen now is we would take a photograph and I would give you a certificate, and then shamelessly we would put it on the Doctoral College website. You will get a certificate at some point, and I really hope that we will be able to do that in person at some point. So thanks again, Rob, and hopefully you'll hang around for the second lecture, and maybe the glass of wine will be, unfortunately, it'll be in your own home rather than with us. So thanks very much, Rob, and again for the interpreters.
Thank you very much indeed.
Okay, so it's time to move on to our second lecturer now. Our second lecturer is going to be Tunde Huszar, and I can see her now in the middle of my screen. So Tunde won the CLS prize. I don't recall the year, maybe it was 2090, probably 2000. She's currently a postdoc fellow at NAST in Georgetown, and she's going to give her doctoral inaugural lecture on breaking ground with new technologies in forensic genetics. Tunde, we're very much looking forward to hearing what you have to say. So I would normally say the floor is yours, but the screens are now yours. So take your time and set up your slides. Thank you.
Thank you very much for the introduction. Let me share my slides with you. And it should. Good. Are you able to see my slides? That's working. Okay. First of all, I'd like to thank you, everyone who's taking the time in this afternoon or evening, wherever you are in the world, to listen to these lectures. And obviously, I would like to thank the Doctoral College, the department, and all those who put me forward to this award, who selected me, and then who awarded me this prestigious award. I'm really honored. And I feel this award is not just mine, obviously, but a lot of people who together we work to achieve these scientific achievements, which was actually together as my PhD. When I was invited, I was a bit worried about 20 minutes limit. Who knows how I talked, and you know that I can. I'll talk to a long time. So what I decided to do, instead of trying to squeeze in my whole PhD into 20 minutes, I decided to take the thread of new technologies, more specifically sequencing technologies, that were common in my PhD and the different projects that were my PhD, and try to give you snapshots of different achievements that we put together during these years I spent in Leicester. At the moment, I'm in the US at the NIST, National Institute of Standard Technology, and also at Georgetown University, working on this very similar project. So I'm continuing what we started in Leicester. First of all, just as a reminder, I would like to just go through quickly what am I going to talk about when I talk about forensic genetics and what markers I used in my project. The markers I'm going to talk about and where these are. So most of them are in the nucleus. These are in the chromosomes, either the autosomes or on the sex chromosomes. What my interest was specifically is the Y chromosomes. So here on this slide, you can see listed them and placed them nicely on their chromosomal locations. All those markers I used. Apart from the markers in the nucleus, I also take a look and studied the mitochondrial DNA, which is outside of the nucleus, inside.
Sorry to interrupt. I don't think your slides are moving along. Are you on your second slide?
Yes.
Okay, we can still see the first, I think. Is that the same for you, Dave?
Are you seeing chromosomes?
I've just got the first slide. Maybe if you click presentation, it might help.
Something is funny. Let's try again. It says presenting.
So it's not full screen or presenting. No, not quite. So it doesn't look to me like it's present. That one, yeah, that one.
That's where I was. So can you see now forensic markers?
Yeah, I can.
Fantastic. And you can hear me. That's the most important.
Yeah.
Okay, thank you. So just coming back to which markers I'm talking about. So mitochondrial DNA is another marker which is found in the mitochondria outside of the nucleus. So these two locations I looked at, and forensic markers I looked at in my studies in the project that were my PhD. Just to looking at the types of markers, so now we looked at where they are. Well, one of the main markers used in forensic genetics is short tandem repeats, or STRs. These are variable by their size, so that's the most important thing to know about them. How many repeat units they are consist of define what type they are, what name they are, what variant they are. We call these alleles. So how we measure these, we measure the length of these markers, define these and associate them with numbers based on the number of repeats. So that's how STRs, these forensic markers, are characterized. Another type of forensic markers, which are the SNPs, or single nucleotide polymorphisms. These are also used in forensic genetic analysis. So these are practically taking a group of people, a population, and compare their genomic sequences and identify positions. For example, here these red arrows point out to schematic positions which are variable in the population. So instead of, as we've seen in STRs, different lengths of alleles, here we have, in this example, two different nucleotides at these positions. They have for two different alleles in the SNPs. Could be more, but in this example we see these. We can also see that the rest of these examples of genomes are exactly the same in the population. So we are looking for these variable positions to find SNPs. So these are the main two markers that forensic genetic identification uses. Let's look at the process. Everybody's interested in
it looks like your slides are not moving, Tunde.
Okay, I have to figure out again. So you've seen the routine.
So we see one that says routine. In order to advance them, if we're having issues, it might be worth advancing them on the left-hand side manually, so that you click down each time. That might that probably would work.
Okay, that's interesting. Let's try that then. Okay. So this was a routine forensic DNA analysis process. So we're starting with a biological material. It could be either from an exhibit from a crime scene, or it could be, for example, here a buccal swab from a reference sample, or it could be even non-forensic, it could be just research material, so donors giving buccal swabs for providing their samples to the project. Either way, whether it's from biological exhibit or donor samples or reference samples, the point is in the laboratory, the DNA needs to be extracted from these materials, biological materials. Once we extracted the DNA, in the laboratory, we have to put through in a process where we actually amplify it, make it multiple copies, and target these markers we just mentioned we are interested in. The reason for this, because we want to identify them, and to be able to identify the signal need to be strong enough for us to be able to do that. So that's why we have this step of amplification, making many copies of the targeted regions. After that, we prepare these amplified markers, and in a classical routine DNA analysis workflow, these are separated by length. So for this, we normally using capillary electrophoresis, and this is just a standard machine where at the end we end up with STRs separated by length, and that gives us a profile. So I'm going to step now to the next slide. Let's see if that works. Are you seeing a new slide? Can I get the feedback?
No, if you click it on the left.
Left, as in.
So just click the slide, and I think it should appear. I would imagine. So you want slide 6, right?
For me, it appeared. So that's why I'm just asking if you are able to see it.
I think you need to click slide. You want slide 6 next, right?
Yes.
So
I guess. I don't know. For me, it's a full screen. I don't see anything else. So I think I could just go like this and then change it and go full size. That's all right. We figure it out. So this is the next slide. So how we change the normal forensic DNA workflow. Well, the first part doesn't change. So the part up until we amplify the targeted forensic markers we are interested in stays the same. But when we're using new technologies, for example, sequencing, and in particular in my project, what I was interested in, massively parallel sequencing, which is like a giant version of regular sequencing or reading the code of the DNA. In this process, we prepare a little bit differently the samples and then allow them for the different platforms with the different process to prepare it for sequencing. So sequencing is reading the code of the DNA. And these platforms I put here are those that are known to be used in forensics, but most of all, the first two are those that are mostly known and used at the moment for forensic DNA analysis. In my projects, I use the first one, which is from the Illumina platform. It's a MySeq, a specific version of MySeq, the MySeq FGX. But also here you can see one from Thermo Fisher Scientific, the INS5. That's another type of sequencing platform. You could also have long-range sequencing with PacBio, or long-range but very mobile, which is on the bottom right corner. That's MinION sequencing from Oxford Nanopore. So these are different platforms that could be used for massively parallel sequencing our standard forensic DNA markers. So the difference, why we want to do this and why we want to use these new technologies when we already have a very well working standard workflow. Well, the difference is in the standard workflow, we are limited how many different markers, how many STRs. For example, we can we can identify from a single sample, a single run. So what we would like to do, we'd like to maximize the data obtained from these analyses. So we don't want to limit ourselves to 20-something STRs. If we could choose hundreds of different markers, and it doesn't have to be only STRs, it could be combined with other markers. It could be STRs plus SNPs. It could be also involving mitochondrial DNA, not just genomic. So there are a lot of versatility. Also, in great numbers, there is the economic cost, which is a lot better per sample in these setups, where we use sequencing reactions millions in parallel. So obviously that helps with the cost. Also, these are very well automated, so that cuts down on work hours in preparation and handling, and obviously a lot more data generated with these approaches. So these are all positives for choosing a better, more robust approach to learning about forensic markers. So let's see what are the DNA profiles, because it sounds a bit cryptic if we are not familiar with it. So as we said, the standard STR markers, when we look at them by length, they are separated by capillary electrophoresis. So we see this profile, which are nice and colorful, and there are little peaks in it. So what we see here, these different alleles, different variants of these markers are separated by their size. So on the x-axis, they are separated, smaller ones on the left and the larger ones on the right. So we can identify a little, okay, let's go back. So we can identify two alleles most of the places, since if we're talking about autosomes, we have two copies from each of our parents, which we inherited. So these two peaks for each of the markers are translated into numbers. So when we talk about the alleles by length, we're actually giving these number strings. Each marker is named, well, these seem random numbers, but actually if we find the numbers after the letter D, that gives us a clue which chromosomes they are on, or sometimes they are named after historically from other genes which they are nearby, and that's how they were discovered. So the DNA profile is actually nothing more than a string of numbers. For each marker that we look at, we have two if we're talking about autosomes. And if you're talking about a male sample, which has one X and one Y chromosome, then obviously one and one allele for those sex chromosomes. If we talk about a female, then we would have two copies, two alleles of an X chromosome marker. So that's what a DNA profile is in a classical term. So when we sequence with massively parallel sequencing, then let's see what are the advantages and what changes. So instead of just seeing two numbers based on the length of the repeat, for example here it says a 16 and an 18, instead of that we are able to actually read the structure of the repeat. So it's not just, oh, we have 16 repeat units. Actually, if we look into the sequence, we're able to see, yes, in total we have 16 repeat units. However, in the middle, one of the units is a bit different than the rest. So we have CTA units in the beginning and at the end, but in the middle, we have a TCTG. So that's extra information. And if we look at other alleles, we can see they have different number of different units. So that gives us a lot of extra information about beyond actually just the number of repeats. And there is a new concept that actually comes with sequencing STR alleles. We're probably all familiar with the concept of heterozygotes and homozygotes. So heterozygotes would be if we have two different alleles at the locus. Homozygotes would be if we have two the same. If we look at this locus here, by length, both of the alleles are 12, so 12 units of repeat. But actually, if we look at the sequence, we're able to see the 12 units for each, but one of them is different. What is noted here is a SNP right next to, in the flanking region of the STR repeat. So we have an extra layer of information here. And this information is not just the STR, but the surrounding region's variation, including SNPs. So combining more than just one type of marker here. And that actual phenomenon is called an isometric heterozygote. Isometric because it's the same length, but actually by sequence it's different. So that's what sequencing by massively parallel sequencing gives us, an extra edge. We have much more information from the same markers, And by this, we actually gain a lot of extra alleles, a lot of extra variants. The other examples here on the slide just shows that these STRs could be very simple or could be quite complex, or even more complex than any of these examples. So just to show you one project of what my PhD consists of, what we managed to achieve by actually sequencing the STR alleles rather than just measuring their length. So what you see here is a list of Y chromosomal STRs, and all you have to just focus on, see the gray parts. That's how many different alleles we have if you just measure the length, and you see lots of colorful things on top. Some has more, some has less, or none at all. These are the extra alleles which we are able to define, measure, analyze if we are using sequencing, massively parallel sequencing on these samples. So actually, we unlock a lot of new alleles, increase the allele diversity at these markers, which, if we talk about autosomal STRs, that means we have a greater power to discriminate between people from the same marker set, thanks to being able to recognize a larger variation. So this was one of our interesting output from the PhD project. Next one. Another thing that we actually managed to gather, and the new side of the STR typing, thanks to the massively parallel sequencing, is since we are sequencing alleles, now we are able to find rare sequence variants. So if we are sequencing a large enough number or a diverse enough set, which actually was our approach in one of the projects, that means we are able to find very rare variants. So for example, in the first example here, that's the marker set. Normally, the sequence of this was known as several different repeat numbers of the GAAA unit. And since the unit next to it, this something bolded AAGG 6 times, didn't vary, it wasn't seen as variable, it was considered non-variable. But actually, if we sequenced enough variants, like here we see few rare variants, we were able to find alleles where these were different. So actually, based on these results, we were able to suggest a change in nomenclature for these markers, because these variable units should also be considered as part of the repeat. So that was one of the great outputs of the projects my PhD consisted of. Another aspect, how else we could use MPS, massively parallel sequencing. We can, as I mentioned, we can use it in empty DNA, mitochondrial DNA. Mitochondrial DNA normally is sequenced itself, but with the classical Sanger sequencing method. So why is this better? Obviously, we can have a higher throughput data, and we can have a lot more information from the same amount of samples. So definitely worth exploring. What we looked at from the whole human mitochondrial DNA, there is this smaller portion which contains most of the variations in the mitochondrial DNA. So we focused, as normally forensic analysis focuses on this region, we focused on the analysis of this region. We used a chemistry which just covers this region with overlapping targets. But with new technology comes new problems. And these overlaps created some bias in the data, which is a bit problematic. So if we have new problems, we create new solutions. So one of my projects was about creating a bioinformatics solution to correcting for a chemistry problem. And actually, this was a really, really great outcome. And the supplier of the kit was really grateful and incorporated these findings to correct for these mistakes, hopefully in future products. So that was another aspect of using massively parallel sequencing for forensic DNA analysis. Okay, another aspect that is really important. It's all great that we are finding new alleles and expanding our ability to type and get more information out of the same markers. But is it useful, right? Because that's always the question, how useful, what are you doing? It is useful if we also provide the frequency data valid for the populations. So actual casework can use this data, can use this information. So what we did in collaboration, we used a data set, People of the British Isles, which covers the British Isles, as you can see in this little map that represents the samples in the study we selected from. And what we did, we applied all massively parallel sequencing of these markers that I just mentioned to these samples. Here is an example where one of the markers listed here the different sequence variant alleles, and also by length, as in a classical separation we would see. And then we could provide either by each region or for the whole British Isles the frequencies, how often these alleles seen. So why this is important? Because, for example, if you think about autosomal STR profiles, it is great if we have an autosomal profile and it matches to somebody, but that on its own is not enough. We have to be able to say how likely this matches and how likely to have another person carrying these combinations, these unique combinations of the markers we just reported. Because without that, the evidence doesn't have a weight. So population frequency data generation is really important. So actually all this data can be used in forensic DNA typing, which is now expanding into sequencing. So that is absolutely crucial. That was one aspect we generated a lot of data. Also, not just autosomal DNA, but for the patrilinear and matrilinear markers, so Y chromosomal and mitochondrial DNA markers, we can also provide these kind of frequencies. But since these are not separate markers, but they are tied together physically, so Y chromosome is one physical entity, and all the Y chromosomal markers sit on it like the beads on a necklace. Are you able to see this slide? Let me just double check if we're able to see. Yeah. So for Y chromosomal markers, these are tied together, so they are a haplotype. So what we can define is the frequency of haplotypes for different regions of the British Isles, and this could be useful for tracing lineages of the Y chromosome. Similarly, we can represent it differently by nice colorful diagrams for the mitochondrial DNA too. So population frequency data, not just for autosomal STRs, so identification and direct casework, but also tracing different uniparental lineages is useful and important. And these can also be used for tracing population history, population structure. Okay, next slide. Just pointing to the future, the last two slides is really about where things are going and actually things that are happening right now. So how even these new technologies evolving further. So one of these, what can we do beyond the standard markers? And that is about phenotyping and estimation of biogeographic ancestry. So for example, visible markers such as hair color, eye color, skin color, the estimation of these visible traits could be obtained using these technologies, massively parallel sequencing, say these platforms. And what these do, these are providing estimates. So these are not black and white answers like in the STRs and SNPs for identity or as we just seen in the previous slides, but it's more like an approximation, an estimation of what is possible as a visible sign, as a phenotype from a sample. Similarly, it estimates which populations, the origin, the ancestry of the sample is most likely or most closely related to compared to the known references. So that's very exciting for, obviously, for providing intelligence in forensic investigations. And just for the end, and probably the most exciting, I believe everybody heard about 2 years ago, 3 actually now, 2018 April, there was a big media story about the catching of the Golden State Killer and how they caught it. They actually used genetic genealogy for forensic intelligence purposes. So actually what they did, they used publicly available direct-to-customer genetic testing databases. In this case, it was the GEDmatch database, where they uploaded data derived from crime scene material. And using this as a bait, they were able to find near or a bit further relatives of the perpetrator, back then unknown perpetrator, and then using reverse genetic genealogy from actual genealogical data, as in records, and other data available to obviously law enforcement, not just lay people like us, they were able to narrow down the possibility of suspects, and at the end they confirmed by surreptitiously taking a sample from the possible suspect, which confirmed actually that that person was indeed who was a serial killer rapist who evaded justice for decades. So what is the newest development on this field? The problem is always that there is ethical concerns, ethical considerations, because this is really walking on a very thin edge. Are we in the right to use public databases, people who are just uploading their data for recreational purposes? So the main changes that happened since this very famous arrest is these databases now only available from those who volunteer their genetic data. So the comparison and the fishing for relatives of unknown suspects is only using people who actually actively consent to this use of the data. So that's a great improvement. And also the problem of losing custody of the materials which are part of criminal investigation was questionable. And now there is a new platform. Well, not really. A platform is exactly the same as we would use for forensic marker sequencing, as I just explained. But now it is able to generate markers that are comparable to these home testing genetic data and can be compared without even leaving the evidence leaving the crime lab. Able to generate a profile that is comparable and able to find and identify distant relatives of unknown suspects. So this is actually a 2 weeks old development. So this new kit is coming out, and that's the newest and latest of how we can use massively parallel sequencing in forensic DNA analysis. And let me just finish with the acknowledgement slide. So again, I would like to thank everyone who put my name forward to this award, who selected me and who awarded me this prize. It's obviously that is the Doctoral College, the College of Life Sciences, the Department of Genetics and Genomic Biology, and over everything else, my fantastic supervisors, Mark Joblin, John Wetton, and Celia May, who were putting me toward this. So this was a great honor, and I was very surprised and very grateful for this fantastic opportunity. Also, I'd like to thank obviously my group members who during the years were fantastic and made it enjoyable these years. Also the facilities at Leicester, so that's Nucleus team for sequencing and also the cluster for computing, and also the partners who were involved with my PhD project. And that would be the end of it. Thank you very much. If you have questions, please let me know.
Thank you, Tunde. First of all, you did an absolutely outstanding job in the face of a few technological difficulties in terms of slides and things. Didn't affect my understanding for sure, and Kelly shared a PDF of your slides anyway, so hopefully everybody's had an opportunity to at least see what you were talking about. This was an absolutely outstanding treatise on this new technology. Does anybody have any questions? I have a question or two. We have time for a couple of questions if anybody wants to ask something. I'll try and keep an eye for any hands, or just unmute yourself and ask a question. Anybody? Any offers? Don't be shy. Okay, so I was kind of thinking, and my thoughts were crystallized towards the end, where you started talking about ethics of having databases, the new technology being able to find perpetrators based on linkage to their families, right?
Exactly.
So I'm just trying to square this in terms of, it almost has a kind of Big Brother feel to it, that depositing my DNA or my family's DNA could lead to the conviction of somebody who is related to me. Clearly, if somebody in my family has done something really awful, then that's fine. But it has almost a sense of Big Brother to it. I wonder if you wanted to comment on that or develop that a little bit.
Yes. So exactly for this reason, there is a limit for what type of cases this application, this forensic genetic genealogy, as this field is called now, could be used. So it cannot be used for like stealing chickens or very minor crimes. Obviously, that would not be right, because that concern, what you just said, would be very legitimate. Are you really going to put an unknown relative of yours into a tough position because of you like DTC testing for yourself for recreational reasons? No. So what they did, they defined the actual crimes that this could be used on. So serious crimes, this meaning murders, usually cold cases, so things that could not be solved by other means. And there are many different ways to solve crimes. So this is really like a last resort. So murders, serial murders, rapes, serious rapes, and serious bodily harms. So there are now two different databases that law enforcement can use for these comparisons. So one is what I mentioned, the GEDmatch, which is like it's trying to compare all possible DTC companies. So it doesn't matter which company you tested, you can upload it here and it compares. So it's like a common pond to fish, bait the fishes. But also one of the DTC companies, Family Tree DNA, allows for their users to opt in, volunteer their data. So law enforcement can use that for this kind of matching. And the same applies. So you cannot use it for light crimes or crimes that are not serious enough to actually justify this, you're right, intrusive way of finding perpetrators. So it's really a balance of what is acceptable and what price we're paying for this extra level of intelligence that can lead to guys who deserve to be behind bars actually getting there. So balance.
I think what you've just described is possibly a PhD for somebody looking at the ethics of deploying this kind of technology. So maybe there's somebody out there who wants to do a PhD in this area. But there we are. Anybody have any other questions for Tunde? Anything else for anybody? No questions. So it remains for me to thank you again more formally and to commend to you, the audience, Tunde, as our doctoral inaugural lecturer. And as I said in the last one, what I would do now is I would stand up and shamelessly have a photograph with you with a certificate, and we put it on the Doctoral College website. We will make sure that you do get a certificate, but that all depends on this unlocking process. So once again, thanks very much, Tunde. Really enjoyed it, and to Rob as well. Really enjoyed the session. We ran over a little bit, but that's not necessarily a bad thing. So thanks, everybody. And I should also thank Kelly for setting all this up and doing all the organisation and panicking when we couldn't see your slides and sending around PDFs and stuff. So I think the session's worked out really well for everybody. And I wish you a pleasant evening, and do have a glass of wine or whatever, and enjoy the rest of your evening. So thanks very much, everybody, and good evening.